Well, it's a pleasure to be here. We just switched back up here, and it's great to have. It's been really interesting conversations. I'm going to build off what David just talked about, and we've talked about data platforms. He talked about ways of bringing data together to make it accessible for many people, and I'm really going to talk about use cases in the electronic health record, and really talk through a couple examples across our own experience at Vanderbilt, and it's going to link a little bit to a network we're involved with called the Emerge Network, and then all of us. So I wanted to start here with some slides just kind of talking about the kinds of data we have in the electronic health record and how much we have. So the biobank we've built at Vanderbilt uses de-identified data from the electronic health record. It's all kind of leftover data. That's the sole source of phenotypic data for BioView. It's all de-identified. We're not able to recontact participants, and it began in 2007. The data points on the screen are transformed by square root divided by 20, so it's a real sort of nonlinear transformation, but it gives you some idea of density of those different streams of data, all for people who first enrolled in 2007 or beyond. We have about 250,000 people in the resource, and you can see that we have data going back decades. We use all these data for studies. The most common thing, you see about 70% of the phenotypes use billing codes, mostly ICD codes, and that's 9 and 10 in our resource. But you see narrative data used almost in everything, the clinical documentation realm, radiology data, ECGs, laboratory data, even occasionally vital signs. Medications are used in about half the phenotypes we do. And each of these get validated by looking at medical record, looking at real notes from providers to say whether or not they have diseases or not in subsets. And these can be machine learning models, these can be Boolean logic and things like that. I'm not going to go into a lot of details of how those algorithms, what they look like, but we know we can get positive predictive values in excess of 90% for most of them. And so we're part of a network called the Emerge Network, and this gets to that last question about federation or not. So Emerge is federated by people. Each of these 10 sites that have EHRs linked to biobanks, decide on a common phenotype. They all go execute. We have a common language, but that looks like a Word document, and we execute that within our own EHR. And then we have a common set of genomic data. It's about 100,000 people, and we also have some additional 25,000 or so sequence data and things like that that we look at. And we share it, and that federation process is actually hard. It's one of the points I want to bring out because each of our EHRs are different, and so there's not a common language across those, which is going to get to a little bit about what we do in all of us. But Emerge has been very successful. Despite that, we've done around 100 different phenotypes that we've executed in different ways and a number of successful genomic studies. So all of us. So we've talked a lot about all of us. Many of you are probably familiar with it. It's the goal of 1 million people who are recontactable, re-engageable participants that share in the journey with us as researchers. It was originally described by President Obama in 2015. The network was kicked off in the summer of 2016, and we nationally launched on May 6th of 2018. We currently have that should say 85K, not 85 full participants. We currently have about 85,000 full participants, and that sounds much better, doesn't it? So we'll have to edit these slides before we go out to anyone. My job will be shortened. The 145,000 have started the process in some stage. Importantly, we really emphasize diversity. We're about half non-white. About 75% are what we call underrepresented biomedical research, which looks at socioeconomic status, rurality, sexual and gender minorities, things like that. It is, I mentioned, it's recontactable. Some of the upcoming plans, because we are very young, we're starting GWAS and whole genome sequencing in 2019. Broad is one of the partners for that, and we'll be leading a lot of that effort along with 2 other genome centers. That we expect research availability through the data platform that David referenced sometime in about a year from now. And a key part of this is participants get data back. And that includes their genetic data, that will include some EHR data, that will include their surveys, and other things, and see how they go across other people. And that has a potential to impact clinical care as they take these data to providers. We have 2 recruitment models. The healthcare provider organizations would be the typical way with largely academic health centers. We also have this direct volunteer network, which is looking at things like Walgreens and other sort of minute clinic type providers, blood banks, and other things like that. Everyone has a common enrollment process, agrees to share their EHR. We have health surveys, some in-person visits for baseline measurements and biospecimen collection, and then the idea of leveraging wearables, sensors, smartphones, things like that. We have a centralized data repository for this, fed by a number of different groups. I think about all the academic healthcare providers. We have about 40 or so sites that will be sending us data. And they're already sending us mostly the structured EHR data across the visits and billing codes, medication, and labs. They're trying to map those into common data models and doing a pretty good job, but this is going to be a journey. It's not someplace we've arrived, and I'm not sure we'll ever arrive. We'll always probably get better. And we're starting soon the process of getting narrative data as well, and are thinking about capturing image data and other kinds of things. Centrally, as a data and research center, we are looking to add other kinds of information, like linkage to federal death indices, getting claims data from other sources, and prescription records. That's a real challenge in our data environment, and these things proceed at different levels. We're still working out plans for that. And then for direct volunteers, we're working with other partners to try to get some of their electronic health record data. That's also a process that we see as a journey. One of these is something called Sync for Science, which is a FHIR-based, fast health interoperability resource-based technology that's built into a lot of the EHRs in the U.S. now, or will be built into a lot of the U.S. EHR vendors, and it will enable individuals to share their data to all of us, and potentially for other resources as well. And it's similar to Apple has an ability to capture with a number of different providers your electronic health records to your phone, and that same technology could be used by other providers over time. And all this data combined with our research collected data will be in what we call raw data repository. We remove obvious identifiers and other sort of transformation to become curated data, and that is what's available for research and will be through common tools. So a couple examples built on top of EHR information. So I think longitudinal phenotypes and pharmacogenetic results and things like that are some of the kinds of things we can do in EHRs. And so I wanted to give one example that kind of crosses the GWAS-PheWAS perspective. And so this was a population that we identified that were on statins and then had residual myocardial events, and identified signals in LPA that were associated with cardiovascular disease. You can see that we were evaluated by looking at manual charts, the positive predictive value in 3 different sites, and you can see that this is resistant to degree of LDL change. So this represents a potential drug target for residual cardiovascular risk, and so we did a PheWAS of this locus, and this relates to some of the stuff Robert Plenge was talking about. And so if you look at, let's see, I'll go over here. So you can see, most of the phenotypes that are identified represent indications are all on target for cardiovascular disease. So with the PheWAS, potentially you could identify adverse effects as well, which potentially would have, pleiotropic effects that could be other indications or adverse effects. One of the things that's interesting over here is you can see it's not quite statistically significant, but in the neoplasm range, there are actually lung cancers that fortunately had the same direction of effect, but not something you would expect to necessarily be associated with the LPA locus. So that is something that wasn't statistically significant, but is close, and maybe with larger sample sizes would be something worthy of investigation. So, and then, beyond a single phenotype, we can think about phenotypes in clusters. So just like we talk about polygenic risk scores, you can also look at phenotype risk scores. And so the idea in this process done by a researcher at Vanderbilt is to take a number of features of disease, in this case driven by OMIM, and map those to EHR phenotypes. We're actually just using the PheWAS phenotypes we've built before and then aggregate them together, weighted by essentially their rarity in the EHR. So it is simple information weighting scheme, and you can aggregate these together. And by mapping all the phenotypes to a common ontology, you can basically just iterate this across all the Mendelian diseases for which we have a schema designed for in OMIM or other resources. And so we've done this for about 1,200 diseases. Here's just an example of what it would look for 4 real cases and 4 real controls of cystic fibrosis. And you can see, you have more red on the cases than the controls, and features like bronchiectasis are worth a lot more in a weighting scheme than asthma, and so that gives you higher scores, as you can see on the bottom, for the cases versus controls. So importantly, we're not using the diagnosis, we're just using the features. So they don't have to be diagnosed with the disease. So then we started to say, okay, let's look at people who are diagnosed, and you can see those with cystic fibrosis as cases that were diagnosed had higher scores based on their features than controls, and we did it across 5 other diseases that were picked a priori, and you can see for most of the rest of them, we saw a big elevation in cases, but not PKU. So PKU is part of newborn screening, and so people avoid phenylalanine and hopefully don't get the features. And what this actually is a readout, it shows you that the cases don't have elevated scores because they generally don't have features of the disease, and is what you expect. And then we looked for novel variants with this process, and across just the people we had on array-based genotyping that were rare variants, and then we sequenced those individuals to verify whether or not they had second variants. And so in this case, we're looking at number of standard deviations for each individual as a single point from what their expected score would be given their age, sex, and race. And so you can see that individuals that were diagnosed for cystic fibrosis had scores that were 4 or more standard deviations above expected, and then a lot of these other variants had very elevated scores as well. And some of these novel variants, cause people to need kidney transplants or liver transplants, and so certainly are having clinical impact in the diseases. And so, this is a way to, I think, read out on things like penetrance, to look at pathogenicity, but importantly is a way to look at phenotypes maybe in a little different way as a continuous number, as a score based on aggregation of phenotypes. So as we think about this coming together, starting with EHRs, but a lot of our modern cohorts and the cohorts we've talked about here, layer lots of other kinds of information on top of it, the surveys, the physical exams, we're talking about sensor data and collection of activity monitoring, things like that. And then, the ability for participants to donate information. So that's a screenshot of my EHR sitting on top of my iPhone, and and through that sharing mechanism. And so participants can donate information in as well, and I think that's a mechanism we're thinking about. And then we can do that for things like discovery, and we've heard a lot of examples of that. And then thinking about aggregation of phenotypes, and we just talked about using machine learning approaches and things like that. Really, all these things I feel like starting with the idea of how can we improve the phenotype picture. I feel, as we just talked about, we've had experiences both federated and centralization, and there's some advantages certainly to centralization of these data, or having common ways that we federate. So the problem with federating and emerge, as I alluded to earlier, is we all have different EHRs. But if we could put those information in the same kind of format and language across them, the federation will work better. So the advantage of trying to bring together common data models on top of this and expanding our phenotypic data, especially from my standpoint, adding the clinical data that we collect anyway, it will enable, I think, new discovery. So here's a picture. Just want to thank a lot of folks represent them and the Data and Research Center team. David's hidden like the second row or so there, and Anthony's in that picture as well, and a lot of the folks at Vanderbilt that contributed as well. So thank you. Anthony.
I was wondering if you could say a little bit about the progress on the Sting for Science, and in particular, a world where people can donate their medical records to research. How do we get there?
I think right now, probably Sync for Science and its derivative kinds of technologies are probably the best way we could have to actually sharing information. There's a pilot underway with Sync for Science. We have a small number of records that have been shared to the DRC right now. It's a small number, but we have a certain number of vendors, which represent larger vendors, that have promised to sort of build it and bring it into fruition. So it's one of those things where I think you have to watch over time and see how it develops. Initial results, we don't really have a sense. We are getting real data, which is a progress. We'll have to see what that looks like over time.